Real-time PCR. Real-time PCR is a technique in which fluoroprobes bind to specific target regions on the DNA. Once the fluoroprobes they bind to specific target regions, with the help of that fluorescence, the amplified product or the quantity of the amplified product can be measured. Real-time PCR is used to measure the quantity of a PCR product. The fluorescence is measured in real time, is detected in a PCR cycler with an inbuilt filter fluorometer. So there is a fluorometer that is present in the PCR cycler. With the help of that fluorometer, fluorescence is measured. It is the method of choice to quantitatively measure starting amount of the DNA, cDNA or RNA. So on one hand, we can quantitatively measure the starting amount of the DNA or cDNA or RNA, while on the other hand, we can also quantify how much copies or how much amplification of the DNA has been performed. So these are some of the real-time PCR machines. This is called as iCycler, which was manufactured by BIO301. Here is a light scanner by Roche, Applied Biosystem, and by Stratagene, and by Molecular Dynamics. So these are some of the real-time PCR machines which are used to quantify the DNA. Real-time PCR, also called as quantitative real-time PCR, is often confusingly known as RT-PCR, real-time PCR, or RQ-PCR. QR-PCR or RT-qPCR are more appropriate contractions. QRT-PCR methods use fluorescent dyes such as cybergreen or fluorophores containing DNA probes such as Tachman to measure the amount of amplified product in real time. So in case of real-time PCR or in case of QRT-PCR, usually fluorescent dyes they are used. The common fluorescent dyes, they are either cybergreen or in some cases it is a fluorophore containing DNA probes. Real-time PCR is commonly used to determine whether a DNA sequence is present in a sample or how much copies of the DNA or RNA is present in the sample.